So now, let me introduce Nathn McDannold, who was awarded the Jolesz Memorial Award last night by Kobe. He's going to tell us about his experience in this whole realm of focused ultrasound.
All right, thank you. I'd like to start by thanking the foundation for this award. It was truly a surprise, and it's a great honor, and I know Ference Jolesz would be very, very happy. I decided today to talk about a research study that we did with looking at the next generation of the brain focused ultrasound device, which is the low frequency system, and our tests with this in a large animal model. I'm not going to dwell too much on this because we'll hear a lot about brain today, but transcranial MRI-Guided focused ultrasound allows you to ablate regions in the central part of the brain. And it's a completely non-invasive approach being used for functional neurosurgery and tumor treatments. As we probably all know, the current limitation of the device is that we can only target very central locations in the brain. And this red curve in the red curve shows the area that Insightec estimates that we were able to ablate. It may have increased a little bit with all of the clinical experience, but we know that it's a huge problem trying to get more peripheral or areas closer to the skull, and that arises largely from heating of the skull limitations. So the one way to help reduce skull heating is to reduce the ultrasound frequency. So you get less aberration and you get less heating. You also can steer the beam more electronically. However, you also reduce the heating at the focus at the same time, and it's not clear how, if we're going to win and be able to get a bigger treatment envelope. It's also not clear if we can really avoid significant cavitation effects, since the cavitation threshold is lower as you decrease the frequency. So we did tests with the 220 kilohertz version of the Exablate Neuro. We did these transcranially in rhesus macaques, and we produced lesions again in the central part of the brain. The monkey's head is very small, so the problems are magnified. But we then ablated areas in the thalamus. The goals of this study were to determine whether we could actually increase this envelope. We were doing that by looking at heating at the focus, and then we compared this to a prior study that was done at Brigham when Kalervo was running the lab at 650 kilohertz with the prototype device of the mid-frequency system. We also wanted to evaluate the cavitation-based feedback to make sure that we don't get out-of-control cavitation. We were also interested in whether a cavitation was associated with any sort of heating, so we looked for what we call thermally significant acoustic emissions, and then we wanted to establish cavitation thresholds in the brain at 230 kilohertz. And then finally, we were interested in whether these lesions were purely thermal or did they have a significant effect from cavitation. We looked at this by comparing the thermal dose with the lesions and looking in histology. So what we found was that unlike with the 650 system, where we really couldn't get much focal heating at all in the monkey, we were able to get a significant temperature rise in the monkey at high enough levels to ablate the tissue. We produced these small lesions that were enhancing on T1. In this animal, we did 2 sessions, once a week, each hemisphere. So this is the first week, this is the second week. And if we look as time goes on, the one from the first week increased in size. This monkey recovered and is still alive, and at 19 months, you can't see it on there, but you can see very small lesions present in the brain. So this was very encouraging to look at that we were able to do this in a monkey where we couldn't do it before. This is another example. Here we had a thermal dose that was less than 240 minutes, which is the value that people typically use for high temperature ablation. But we did see BBB opening in the thalamus, nothing on T2 weighted images. This monkey was sacrificed at 24 hours, and we imaged the brain post-mortem. And so we see this delayed effect where we see this significant lesion in the brain that was not there when we first ablated. So, and the area in which the thermal dose seemed to match agreed with about 30 equivalent minutes at 43 degrees, and that 18 minutes at 43 degrees, which was a threshold that we chose based on work that we did a long time ago in rabbits. So this was similar. So with heat, we can disrupt the blood-brain barrier, but at least from what I've seen, you always get a lesion afterwards. We did get some surface damage. This is that first monkey that's still alive. Here we produced some damage. It's probably not in this case in the brain, but in the dura. When we went to higher exposure levels, we did get damage in the brain. But that was not unexpected because the size of the monkey head is very small. The area is about 50 square centimeters compared to over 300 for a human. So now looking to see if this was really a thermal effect or not. Here I'm plotting the acoustic energy and the temperature rise. And this is for the first 3 monkeys. And we see this nice linear response. So typically if we have cavitation-enhanced heating, at some point we see a big increase in the temperature rise. And so at the focal point and on the brain surface, we see a nice linear response from between temperature or the acoustic energy and temperature. This was the first 3 monkeys out of the 4. I should have stopped here because we did one more and we get a different result, but it was also very linear at the focus. Based on this, we could now compare this to the study that we did in the past with the mid-frequency system. Here at 220, we were able to get higher temperature rise per energy. At the focus, this is in the first 3 monkeys, this is in the second, and at the higher frequency years ago, we didn't see any heating at the focus. And at the same time, the brain surface was heated much less. Here we're looking at the hottest points on the brain surface, and here we're looking at a sort of an overall average. So it does seem like this low frequency device in principle should significantly increase the treatment envelope of doing focused ultrasound. Looking at histology, the lesions are completely consistent with what we've seen before with thermal lesions. Here, this is a higher temperature lesion in the case, and the red contour are the dimensions of the dose contour at 240 minutes. So this high temperature lesion that we produced does agree with roughly with what we've been using as a guide. But this yellow contour, if we look closely, we can see that the neurons out here are dead or dying. This was 2 hours after the ultrasound. And we can see that is also seems to agree fairly well with the lower dose threshold of 18 minutes at 43 degrees. This was another monkey where we did 2 days after sonication. This is the one where we didn't get 240 minutes. And you can see again that the area of the ablation agrees with the 18-minute contour, and the area of the lesion, all of the histological evidence of the lesion looks completely thermal. And in fact, in this case here, with this lower temperature lesion, areas around blood vessels were still intact, which is probably from a heat sink effect. We also monitored cavitation and tested the real-time controller, which was based on subharmonic emission and wideband emission. In general, we did see subharmonic and wideband in most cases, actually. So it suggests that there was some cavitation activity occurring. This was done with a detector. We don't know exactly where the cavitation activity occurred, but it was able to use this real-time controller to rapidly control the sonication and stop it instantly if there was any effect or larger cavitation signal. And we didn't see the evidence of cavitation-induced damage in the histology. So here, if we plot the temperature rise and this cavitation at the subharmonic and in wideband, we do see as the temperature rise increases, we start to see an increase in cavitation activity. And if we look at the threshold for this, we're calling thermally significant cavitation, which was 3 times higher than the lower power level sonications, we do see that there is a threshold effect, and this is the regression that we used. In the first 3 monkeys, at least, we did see also a threshold evident when we looked at the acoustic power. For some reason, in monkey 4, we did not get the significant cavitation activity. But from this data here, which is a bit messy, we can calculate cavitation thresholds. I'll come back to that. We also simulated all of these sonications. This was, I won't go into the details, but we used a K-wave pseudospectral finite difference time domain method to simulate the pressure amplitudes. This included shear propagation, and we also then used the bioheat equation to estimate the temperature rise. This was earlier work that we published earlier this year, I believe, and showing that these simulations that we did, at least in the near field, in this case is here, we had a very good agreement between the simulations and, in this case, BBB opening. So we think these simulations are on the right track. This is an example of the simulated heating that we have in the monkey. You can see that in the focal area, they were very similar. On the brain surface, the levels were similar, but the locations of the maximum were often very different than what we observed experimentally. And this may be due to some limitations of our simulations. So this is a plot showing the measured temperature and the simulated temperature. And we did see a pretty good agreement. Everything seemed to correlate with each other, so I don't know how much to read into this. Also, the values that we used for the simulation, the literature values vary significantly, so we did have the ability to sort of move this curve up and down just by choosing what parameters we wanted to use. But one of the things I wanted to point out was when we looked at the difference between the measurements and the simulation as a function of the temperature, a measured temperature, in the areas where we had cavitation, the simulation or the measurements were consistently higher, and the simulations did not include bubble activity, so that we think that this maybe supports the idea that we did have some cavitation heating at the focus, but it was very small. From this, we can estimate the threshold for cavitation is about 2.2 or 2.1 megapascals, and it compares roughly with what I was able to find in the literature. So overall, it seems like this low-frequency device can increase the treatment envelope. This seems like these lesions were thermal and not cavitation. We were able to control the cavitation in real time. I think we need to look at this in tumors, and thermally significant cavitation was detected in 3 of the 4 animals. The source of this is unknown. In the past, with cavitation and enhanced heating, there's a much larger increase in heating than what we were able to see here. And we did see a good correlation between simulation and measurement. And one thing that might be happening is when we have cavitation enhanced heating where we don't have this big jump, perhaps we had Vesselspasm that occurred during the sonication. And this has been showed, this is from Kalervo's paper from about 20 years ago, and this was from a BBB study with injected microbubbles. So if you have microbubbles, you can induce vascular spasm. And I think in the one monkey where we didn't see cavitation and we didn't get as good of heating, perhaps we didn't initiate this vascular spasm. And this could perhaps increase the temperature a little bit compared to what we would see with cavitation-enhanced heating. And with that, I would like to finish, and I would like to acknowledge all the people who worked on this, and again, thank the Focused Ultrasound Foundation for this really great award. I also wanted to point out the support of this was, this is probably the last study we did as a joint project between my lab and Kalervo Heinenen's lab. So this has been a very long journey, and I'm very happy to be able to share this with you today. So thank you.